So an overview of cellular respiration, we're going to look at the chemical equation, and what you see are your reactants on the left. You have glucose plus oxygen, which is what you're breathing in, and it's going to go through this series of glycolysis, citric acid cycle, chemiosmosis, and electron transport chain. And ultimately, at the end, when the sugar is completely broken down, you're left with 6 carbon dioxide molecules and 6 water molecules. So if you look at glucose as a sugar, it is a monosaccharide sugar, and you can see that there are 6 carbons here, 12 hydrogens, and 6 oxygens. And then you have 6 O2, because breathable oxygen is a molecule of O2, two oxygens bonded together. The role of cellular respiration is to completely disassemble your sugar. We want to break all of the bonds. Those are covalent bonds, shared electrons, and so we want to break all of the covalent bonds and release every single one of those carbons as a CO2 molecule. Okay, that's the whole point of this process. In the lab today that we're going over, the cellular respiration lab, we want to investigate 2 hypotheses. So in your lab manual, if you have your lab manual, there's 2 hypotheses. One of them says, although both plants and animals undergo energy demanding processes, animals are more active than plants. As a result of their high activity levels, animals may have mitochondria, may have more mitochondria that are used to support higher rates of cellular respiration. So what we're saying is, just because of the metabolic behavior of animals, that they can move around and crawl like the worms versus the bean sprouts, which don't crawl, there's going to be potentially more mitochondria in the worms than in the beans, and therefore maybe a higher rate of cellular respiration. The second hypothesis addresses temperature. So hypothesis 2, temperature increases collision and reaction rates between molecules and therefore increases the rate of chemical reactions, including cellular respiration. So that's why we're running the experiment at 2 different temperatures, 23 degrees Celsius, which is going to be your room temperature, ambient room temperature, and then around 15 degrees Celsius, 13 to 15, depending on where we get the water, to see what happens to both the bean sprouts and the giant worms when we lower those temperatures. How does that affect the rate of this reaction by measuring your CO2 output? Okay, the greater the output of CO2, the faster the breakdown of the sugar. So the more CO2 you have means a higher metabolic rate. If we switch the temperatures and we're using the same exact organism and we see that we get varying amounts, well then that is an indicator that that second hypothesis holds true. So this is why we want to look at, again, we're looking at 2 hypotheses. The difference in metabolic rate, the breakdown of sugar to produce CO2, the rate of cellular respiration in mung beans, which is a plant, versus giant mealworms, which are animals. And then we want to take both those organisms and run them at room temperature and cold temperatures to see about the second hypothesis, which is going to be whether or not there is a significant effect of temperature on the rate of cellular respiration. So I'll be running all 4 treatments, room and cold for worms, room and cold for the bean sprouts, showing you the data. I'll be collecting it on the LabQuest units, and then we'll be completing the lab tables together. So I will produce another video on cellular respiration with a handout that I'm going to attach to your Canvas page so that we can talk a little bit more about this complete total breakdown, which occurs actually in the citric acid cycle of cellular respiration. And so we'll get to that later on.